Okay, so the full title of this webinar is New Variation Services for Normalizing, Remapping, and Annotating Variants. As I said, I'm Peter Cooper from the customer service part of NCBI. You can certainly write to me, peter.cooper@nih.gov. Lon Fan is also here with me, and he's the head of DBSNP. And Rana Morris is also here to help with the questions and any technical things that we have with the webinar. So what we're going to talk about today are these sort of bullet points here. What are the variation services APIs? We're going to talk about the variant model that that these APIs use. It's called Speedy, SPDI, and I'll spend a few minutes talking about that and explaining how that works. And then we're going to talk about some applications of the Speedy variation services. I'll show you what you can do with them, and I'll just show you an example where you can see them in action here, although they work behind the scenes here in a lot of ways. I'll show you a couple of interfaces that you can go to sort of test them out, to play around with them, to see how the things work, to show you what kind of results you're going to get back. And then I'll do some live practice using those interfaces, and I'll go to the command line as well and show you a demonstration script that will show you how to access them using a script. And then, of course, we'll have questions at the end. Okay, so what are these things, variation services APIs? This is a set of URL-based or RESTful programming interfaces for accessing and working with variation data at NCBI. As I said, they rely on this new variation representation called Speedy, which stands for Sequence, Position, Deletion, Insertion. Variation services and Speedy are used internally at NCBI in DBSNP and ClinVar, and it's how we manage to deal with these large numbers of variants that are present in both of those resources. Okay, so I'll talk a little bit about the variant model for the next few minutes. It's a simple representation that uses a sequence ID, that's what the sequence part is, the location, and the alleles. A deletion, you take something out, an insertion, you put something back in. These are going to represent variants with precise coordinates. They're much easier for variant normalization, and we'll talk a little bit about what I mean by that in a few minutes. They're designed to be machine-readable, although people can figure out what they mean as well. Let's explain why they're a little more machine-readable than people-readable in just a minute. They're amenable to high-performance computation, which is why we use them internally at NCBI for sequence variant processing. And you can represent both nucleotide sequences and protein sequence variants. At the bottom of this slide, there are some URLs that will take you to a web document about Speedy Notation, which is helpful. But much more helpful and much more detailed is the preprint that's available on bioRxiv, and there's a link to it there on the bottom of the slide. So if you want to know more about this variant model, that's the place to go to find out all about it. Okay, so here's an example of a variant that's shown in Speedy Notation, and this is sort of the linear form of this. And as you can see, also on the slide, there's a structured format, a JSON, a JavaScript object format for the Speedy, which is what the APIs are going to return to you. Basically, you have a sequence ID. This is a reference sequence for chromosome 8, a position in that reference sequence. Then there is a base that's deleted and a base that's inserted. And notice that the Speedy variant model doesn't require that you have the deleted base there. You can just use the length of the base. And that brings me to the next point on this slide, which is that Speedy uses, and this is the part that makes them a little tricky to read for people, it uses a zero-base coordinate system, and it's an interbase one. So basically, the insertion deletion starts between bases 198, 1972, and 198, 1973. It's the space between those bases. So the G that's being deleted is the G at position 198, 1973 in the one base sequence coordinate system. And so the advantage of this from a computational point of view is that you can actually represent a pure insertion with a variant length of 0. And here's the corresponding example of a Speedy notation for a protein variant. Again, written both ways with the deleted sequence base or the deletion length. And again, you can get this as an object, a structured format from the API. Now we use Speedy behind the scenes and the variant services behind the scenes in the variation services, variation databases and resources at NCBI. If you want to see them live and in person in action, here's a place where you can see that. The variant search in ClinVar uses Speedy. And so if you've searched ClinVar with HGVS nomenclature recently, you'll see these kinds of things. It'll let you know that you might have a variant that's not in ClinVar, and then it will map you to DBSNP, or there might be an equivalent variant that's present in ClinVar, and it will link you to that variant. So what can you do with these variation services? Sort of three things you can do. You can normalize your variant, and that means finding the equivalent one in NCBI, DBSNP, and ClinVar variants. Transform your data, so convert formats. So if you have a Speedy, you can get the HGVS and vice versa. And then you can do all these kinds of things to standardize the way your variants are shown. You can correct for left and right shifting for insertions and deletions. And we'll talk a little bit more about that in a minute when we talk about contextual alleles. You can transform your data into right-shifted HGVS or left-shifted VCF, which is a preferred way of writing them. A very useful thing you can do is to remap your sequence variant. And so that uses our alignment database system to allow you to map variants between assemblies, between various kinds of sequences that are in there. It also gives you access to annotations because you can access the SNP records in a structured format, and those records are going to include functional consequences, allele frequency, clinical significance, and all kinds of other important information about the variant. A couple of terms that we're going to come across today that I'm going to mention. One of them is the idea of a contextual allele element. I'll say that one first. These are sort of alphabetical on this slide. So a contextual allele is a unique normalized representation that is corrected for something called over-precision. And over-precision is pretty easy to understand if you look at a portion of DNA, for example, that has a low complexity region or a repeat in it. And if you look at these examples shown at the bottom of the slide, we can think about a deletion, and we see those GA repeats there. If I remove one of those GAs, it doesn't matter which one I remove, I'm going to wind up with the same sequence result. And so really over-specifying which one of those I use is what's called over-precision. The problem with that is it can give rise to things that look like different variants when they're in fact not different variants. And it's one of the things that Speedy has been very helpful for because it corrects for these kinds of things. The other thing that I want to mention is this canonical representative allele or representative record that you can see. And this is a really useful thing to do with the variation services. It'll allow you to map to a particular sequence model. And the thing that we call the canonical allele is usually based on the latest genomic assembly, such as GRC H38. So if you wanted to map a allele on a different sequence to the standard allele in GRC H38, you can do that using the variation services. So this is just a slide that shows you how you can do these kinds of things in sort of a schematic. You've got all these different variants in different formats. You convert them to a Speedy. In particular, you want to try to get the contextual alleles, and those are going to normalize for overly precision, overly precise variants. Correct, and you can correct all your problems to make your VCF and your HGVS compliant with sort of the standards. And then you can also remap these using the alignment database system into various genome builds and things like that. So here's a couple of simple examples of sort of normalization or standardizing your variants. So at the top of this slide, I have an HGVS name for a variant, and it's written as an insertion, so insert 2 T's. I can convert that using the Speedy services to a Speedy contextual allele, and this is an unambiguous representation of this particular variant. Then I can convert that back into HGVS on all these different reference sequences, and the preference is to make this represented as a duplication rather than an insertion. And there you can see the correct way or the preferred way of writing these as a duplication. This is a more complicated example. First of all, you'll notice that the HGVS expression contains a GenBank accession number here, and that's the BRCA2 mRNA. because the alignment database system has some of the INSDC or GenBank sequences in it aligned to all our other reference sequences and things like that. And so this is written as a deletion, and it's actually a left-shifted HGVS, which means that the deletion is shown sort of at the left-hand side of where this variant could be, because this is a problem because this is a repeat sequence. I can turn this into a unique representation called a Speedy contextual allele, and I can normalize the HGVS that way. And so then I will get back to a right-shifted, so that's shown at the right-hand end of that insertion deletion. And I can, of course, get back all the different contextual alleles and all the HGVS expressions. And, of course, I'm mapping it to chromosome 13 here in this way. Okay, so how do you do this? If you were doing it with a program or a script, you could use the API, which is a URL-based system. There's a base URL that's written for you there. The services you can use that take various kinds of input, they take Speedy, they take HGVS, VCF, and you can get back the RefSNP records using this beta RefSNP service. And there are some example URL calls. You can try those out yourself right in your web browser if you want to. And I won't click on them here because we'll do some live examples here in just a couple of minutes. One thing that one of the developers wanted me to point out is that the V0 URLs will be stable and backwards compatible, so you don't need to worry about changing that. So here's a full list of the services and functions, and these would be the things that you would add on the end of that URL. There are two demonstration interfaces. There's a simple web demo and an API documentation interface, and I'm going to show you both of those and show you how they work. The simple demo interface, which is at this URL here, just go to variation/services/demo, and you can put any HGVS expression in there you want. This happens to be the one for the fairly famous CFTR Delta 508 deletion. And I can take that single HGVS and I can do all the things that Speedy services do for the most part. So I can get the Speedy representation. I can get the identifier here that will give me the link to the RefSNP record. I can get the normalized HGVS, which in this case is the same, and I can map it to all these other sequences here that are available. The other interface, which is useful in particular if you're going to start writing software that accesses this, is the Swagger documentation of the API. And it lets you see the full API response, and these are the various services here. You can expand these, and you can try them out. So here's an example where I can put in the HGVS like I did before, and I can run this to try it out and get back the Speedy, the same one that I got a few minutes ago on the simple interface, but now it comes back in the structured format. And so it's going to show you the header and everything else. It also gives you the request URL that you could use to get this, and a curl command line. So if you wanted to try this out on your command line to test it out, you could do that. Okay, so just to wrap this part up, the main point here is, you know, these are demonstration interfaces. You're really going to want to be accessing these using software. And LonFon has put up some tutorials up on a GitHub site, the NCBI GitHub site, and that's the URL that will take you there. There are a bunch of tutorials here about how to manage the JSON format from dbSNP. There's also a set of variation services demonstrations here, including a Jupyter notebook that shows you how the calls work, and a real simple script in Python that will let you access things via the API. Okay, so what I hope we have time to do now is to try to do a couple of demos. And the first ones I'm going to do are going to be using the variation services. We're going to the web interfaces. We're going to try to decide if two variants are the same. And then I'll show you the Python demo script from GitHub, and we can show you how that runs on the command line if we have time here. So let me go ahead and back out of my slides. And I'm going to go over to a web browser. And what I want to do is to go ahead and take the two variants that we showed in our slides a few minutes ago. And if I have made myself a little cheat sheet that's available on the FTP site. And so the two variants that we're going to use are here. So here is our first variant, which is written as 27 dup G. Notice that I can also write this as an insertion, which is the not preferred way. And so I'm going to use that one when we run this. The other thing I have is a VCF line here that has another variant that looks quite different. What we want to do is to see if these two things are the same. So what I can do here is go to the simple interface here, paste my VCF, and run this. So it gives me the speedy representation. It gives me a link to dbSNP. This is a variant that causes beta thalassemia. There's a normalized HGVS, so it turned that insertion variant into a duplicate variant, which is the way we prefer to have them written. And here's it mapped over to all the other sequences that we have available here. Now, what we want to do to find out if these are really the same variant is we want to map them to that canonical representative. And that's going to be this one here if we turn it into a speedy. So I'm going to go ahead and do that. I'm going to grab that and put that up here. And so this is our speedy representation of that particular variant. Okay. Now, what we want to do is to see if we get the same speedy representation for our HGVS allele. And so what I'm going to do is now go to the variation services Swagger documentation, which is over here, and we're going to compare the representation that we get for that allele that's shown in the VCF. So let me go over here. I can expand any one of these I want. For example, if I wanted to try to get that to run that HGVS expression to get the speedy, I could do that here. And that's in my demo if you want to look at the script, but I'm not going to do that just to save some time because we've already done that and we've seen how that works. So what I want to do is to get the contextual allele from the VCF that we have here. So let me get that open. So what we want to do is try it out, okay? So we want to put a chromosome. Now you don't have to have the accession number here, which we don't have. So the thing that we're working with is over here. So we've got chromosome 11. Sorry, wrong one. We need our position. We need our ref, A, and our alt, which is AC, and I think I can remember that. Let's see if I do. Now, what we don't have is the correct assembly here. We need to know the assembly because we need to know, all we have here is for chromosome 11. And so the assembly that I want to use is 25. Let me verify that. That's the one that was in my notes over here. Yeah, so that's 140525. And so there's my speedy. It's returned to me as a JSON, but it's the same one that we found before. So these two alleles are the same alleles. So we verified that by using speedy. And that's the same kind of thing that we do here at NCBI to verify these. Okay, the last thing I'm just going to touch on is to show you the GitHub site, and maybe we'll run one or two examples from that on the command line. So let me go over there. My cheat sheet, I have the GitHub site here. So here are the tutorials that are working with the dbSNP JSON. Here are the variation services that we can use today to do that. And notice that there's a Jupyter notebook. There's also this Python script. It's a simple Python script, but it gives you a good starting point if you want to write your own little script to access variation services. And there's also some demonstration files that you can do to run this. So I'm going to go ahead and load the Speedy batch Python here just so you can see the code. And the reason is because it's got these nice little command lines that are just things that you can run as an example. So I'm going over here to terminal window, and I've got Speedy Batch Python in here, and I've got some other files that I can work with. So let's do a couple of things here. So I have a VCF file in there. So let's do that one. And this is one that I didn't demonstrate on the web interface, but it's a pretty handy thing to be able to do. So here's a VCF, and it's, just so you know ahead of time, this is mapped on GRCH37. But what we don't have is an ID column there. And what we can do with this particular part of the script is we can go ahead and put the RS IDs in there. So here's a command line that will do that. And I'm just pasting these in so you don't have to watch me type. Okay, so now we can see we have RefSNP identifiers. Notice that a couple of these variants do not have RefSNP identifiers, and it lets you know that, which is a useful thing. And so you could go ahead and try out some of these other examples if you want on your own. That's just a simple one, and so you can take any kind of file that you want, and I've got those available, and I'll make them available on our FTP site if you want to do exactly the command lines that are in my handout here. And included in there is a way to download the JavaScript objects and things like that. But I think we should stop at this point and see if there are any questions that we need to answer. I think we probably have a few of them. And I'm going to let Rana, Rana's been sort of collating the questions, and we can have her read them and then we can answer them.
Hi, there are a whole bunch of questions. We'll take all of the questions, relevant questions that have come in, create a Q&A document, and over the next week, make sure we have detailed answers to every single question that's come in. And we'll post that in the FTP site that I actually chatted out to you. The first one I think is kind of important for all of NCBI variation services at this point. One of the people specifically asked about using this particular service with non-human genetic variation data. In particular, he was interested in dog. I'm going to let Lon answer that question. Lon Fan.
Okay. So as you know, we NCBI no longer support non-human organism variation, but we are also looking at refactoring some of these services, like the remapping service, and that's a separate function essentially, or utility. And so presumably then, you know, you could just provide any type of alignments data set and it would work for any organism. So if that's something that interests you, when we get your feedback from the survey feedback that you will send out, just comment on there, you know, what you would like to see this work for. Go ahead, put your wish list on there, and then we'll consider it and prioritize with our other tasks or our other workload. So thank you.
Next question, actually, I'm going to combine two. Somebody asked whether or not this service is actually open source. Are there any licensing or use requirements? And another person asked, what are the volume scalability limits of these services? What kind of batches can be used? Can you do hundreds of thousands of variants as an example? So 1, open source, 2, licensing, and 3, use requirements.
Yes, this is all open source. There's no license required. We just have the basic disclaimer of NCBI data and that. And in terms, and what's the other one? The volume. Right now, like we're still sort of in the testing mode. So go ahead and try it out. And we'll have some of the questions regarding volume in our survey. Again, response to that, and that will help us plan in terms of scaling up if you need it. Thank you.
Another user asked, can these services be run on private servers or infrastructures?
Again, we are considering having some of these things run offline on your, so as I said, you can run your own data set, alignment, stuff like that. Something we look at planning in the future, refactoring a lot of these functions that is run on as a utility offline. Again, if you can just respond to the survey, there's your wish list on there. Okay.
Okay, now we're going to get a little more into the data. One of the users said the RefSNP service is listed as still being in beta testing mode. When will the final version be released?
The RefSNP page is actually live now. It's actually the final version. I think the alpha label has been removed on that page within the last week. Thank you.
So it'll still be in development, continuous development.
Yeah, exactly. We'll continue to improve based on user feedback. So if you got anything you want to, you just go ahead and write the help desk and we'll try to prioritize it.
Okay, someone is really interested in mapping alleles over to the ClinGen registry alleles using the contextual alleles. Is that possible? And I think he may want to discuss ClinVar at that point.
Yeah, so right now we're not integrated with the allele registry yet, but I think it's something we could discuss with their developers. Right now they already integrate DBSNP data. I guess it's possible they could, we could provide all the contextual alleles, and then it would make it available for you to normalize it with the allele registry alleles.
Now formatting. A user was interested in posting a Speedy JSON representation. Is that possible for posting in JSON for mapping purposes?
So if you go to our API, the variation service page, some of the functions will support post function, others will not. Again, if you need other functions to post, again, just put it as a feature request in our survey, and again, we'll look at it.
Peter, you may have a slide that you can show for this. Now we're talking about VCFs as an input file. And they wanted to know if you're going to put in a VCF input file for, let's say, a Speedy batch, how would you specify the build itself? Let's say they want to actually do a search on build GRCH 38 as opposed to 37.
So in the you have to pass I think, and Lon can correct me on this, but I think you have to pass the assembly accession number.
Right. There is a parameter for you to pass the assembly accession number, so that we'll know which assembly you're referring to for each of the chromosome.
Yeah, in that example, I didn't point it out, and I meant to. Let me see if it's still there. Let me escape from this for a moment. It should be still there. And I think on that example, it should show you the URL, and I should have pointed that out. This is the newly released site, so it looks a little different. But if you see this URL here, where I passed the VCF fields here, so here was the chromosome 11, there's the position, there's the variant. You're using this service that gets you the contextuals. We're passing the assembly as this assembly accession number, and so you would need the assembly accession number for GRCH 37, which this one happens to be the assembly accession number for H37.
Yeah, if you look at the Speedy batch Python script that's on GitHub that Peter demonstrated, if you look at the codes, it's the version that's passed in as an example in there.
Right. In fact, that's the example that I ran. In that particular case, that Python script is fairly simple, so it has the assembly accession encoded in it, but you could easily modify it so that you could give it the assembly accession number.
All right, it's after 12:30, so there are a couple of other questions that are very, very specific and technical with regard to the Speedy format, and they're interested in understanding the rationale behind some of that. We will actually write that up in the Q&A document so that you have a better and a fuller understanding of that.
So the bioRxiv paper that's linked in that slide explains a lot about the Speedy rationale and details about how it works to make those contextual alleles. So I suggest you check that out, because that may answer your question. It depends on what it is, but we'll definitely get the answer in the document that we put up. Okay, so I think we're gonna wrap it up, and thank you all very much for coming. We're gonna go ahead and sign off now.